Hi everyone, welcome to Basic Science Series. I'm your host Lokendra Kumar, and today we're going to design mitochondria. We're also going to discuss the functions of these individual components that you're seeing on the screen, right? I have already designed the individual components. I'm going to assemble them and make their structure. While I'm doing that, I'm also going to discuss the functions associated with these specific structures. I hope that this video is going to help you to understand mitochondria well enough so that you can study in detail about this particular organelle. Alright, so with that note, let's start the designing process. But before starting the designing process, I do want to mention that mitochondria were first described by Richard Altman, and Richard Altman described them as bioblasts in 1890. After that, Blenda in 1897 termed these organelles as mitochondria. So that was the brief history of this particular organelle. This is the most important organelle because all the cells, they require energy, and this is the organelle that is known to provide the energy, and it's also known as the powerhouse of the cell. Alright, so now I'm going to jump on to my next slide where I have these individual components separately designed, as you can see here, right? So what I'm going to do is, let's start with the outer membrane. So here you can see I have this outer membrane, and it looks like a shell, right? And the upper region, just consider it like it's cross-sectioned so that we can see what is inside the mitochondria. And mitochondria, what are mitochondria? They are basically oxygen-consuming cellular organelles, and they are floating inside the cell. They play an important role in the energy generation processes. So here, this is basically the outer membrane, and it resembles the plasma membrane, phospholipid bilayer membrane of the cell. Has the similar structure. After that, you have the inner membrane. So let me bring this structure on top of this here. So you can see that the second structure, when it's on top of the first structure, you can see the folding of the membranes, right? Something like that. So the second structure is showing you the inner membrane. So you can see here these foldings, they are known as cristae. So this is the inner membrane, and these foldings, they are present towards the inside, and they look like fingers. So finger-like structures are the cristae here. Before I move ahead, let me label these components. So here, mitochondria. So I'm going to label this mitochondria like this. Okay. And the second is the outer membrane. So outer membrane, I'm sure that now you know what is the outer membrane. So this is the outer membrane. And then you have the inner membrane here. So let's label this as well. So here you have the inner membrane, and then you have the space between inner membrane and the outer membrane, right? And this particular space is known as the intermembrane space. Okay, so this is the basic outline of the mitochondria here. And then you have this dark colored region, which is matrix space. And I will discuss the function of these one by one. Let me first label this structure, which is the matrix space here, right? So before I move on to the specific parts, I do want to mention that the mitochondria, they are known as the powerhouse of the cell. And some of these cells, especially sperm tail, muscle cell, liver cell, they have huge number of these cellular organelles. For example, in case of liver, you have the process of detoxification going on, so you need a lot of energy to do that. So you have high number of these mitochondria. Usually you have 2000, around approximately 2000 mitochondria per cell, 20% of the cell volume. Next, the important point to remember is the inner membrane is rich in many enzymes, coenzymes, and other components of the electron transport system. We're not going to discuss in detail about the biochemistry of this organelle here. I'm just gonna focus on the structural components and their functions. So you have these individual components here, and now you can see our structure is getting there. Now let me label the cristae. I hope that you remember what is cristae. Cristae is these finger-like projections of the inner membrane. And then you have the DNA molecule. So this is the DNA molecule. I'm going to place it somewhere in the center, and it is mitochondrial DNA. It's different from the cellular DNA. So mitochondria has its own DNA molecule because it requires energy, it requires proteins to carry out many functions. So it has its own small genetic information stored inside the DNA. It's known as mitochondrial DNA. And further, to make the proteins, you need to have ribosomes. So I also want to discuss the function of ribosomes here. I'm sure that you all are aware that ribosomes, they play an important role in case of the protein synthesis. And these are the mitochondrial ribosomes. So let me distribute these ribosomes. So we have an important organelle, which is the mitochondrial ribosomes. We're gonna label these ribosomes, and we're also gonna label the DNA molecule. So here, let me label it. And we all know the function of DNA is to store the genetic information. Next, you have these interesting proteins. They are present on the outer membrane. They are also known as porin proteins. Porins, right? They are associated with the pore. So basically, they are present in the outer membrane. They work basically in the transportation of the small molecules in and out from mitochondria. So as you can see here, I have the porin proteins. Maybe I'll make another one here and make it a little smaller so that you can see that porin protein is embedded in the membrane. So you have these porin proteins here. Next important molecule, which is this F1 particle. This is important. Let me increase the size, and you can see the size of this molecule is like a tennis racket. So this molecule is important. It's known as F1 particle. It has ATPase enzyme activity for the ATP synthesis. And this structure is present on the inner membrane right here. And it is distributed throughout the inner membrane. So it's gonna take some time for me to make all these structures on these cristae. So let me design this particular structure on the inner membrane, and then we'll discuss these rest of these structures. All right, as you can see here, I have tried my best to distribute these F1 particles on the inner membrane foldings, which are also known as cristae. I still have some spaces that I haven't covered, but you can imagine, right? Everything is covered with these F1 particles because they have the molecule, which is ATPase enzyme, that is required for the energy production. So they are distributed, they are present in high number. That's why this particular organelle is able to produce a lot of energy, a lot of chemical energy. And cristae, the number of cristae, basically they vary cell to cell, and their shapes also. This is also another point that I wanted to discuss. And you also have these ribosomes, which are 55S, which is different than the cellular ribosomes. So you can see here, it has everything that makes it a kind of a separate cell. It has its own DNA, it has ribosomes, it can produce proteins. So it's a interesting organelle, and it's very, very important cellular organelle, and because it produces the energy, right? We have already discussed that. Now we have described the porin proteins already, so let me label the porin proteins here. These are the porin proteins, and obviously this organelle is going to need small molecules for various cellular processes. So these porin proteins, they play an important role in exchanging those small molecules from the outside environment. And then you have granules. And let me distribute these granules throughout the cell. So our mitochondria looks complete. I think we're getting there. We have designed almost all the components that we require, and these are the granules. Let's label the granules. Maybe here I can put the name here and label these organelles as the granules. So we have mitochondrial ribosomes, cristae, matrix, and then polysomes. Since the protein synthesis is an ongoing process, so you will see these polysomes, basically ribosomes, bound to the mRNA molecule. So these polysomes, they are also there. So let me also label the polysome here. Now F1 particle, right? We have already discussed the F1 particle, blue colored tennis racket shaped particles. They are present on the cristae, which is the inner folding of the inner membrane. So now I'm going to delete this. So as you can see here, we have the structure of the mitochondria, and let me center it out. You can see individual components, and if you want to design mitochondria, if you want to make a project and make a 3D structure, then this is how you can basically make the structure, and you can also make a model of the mitochondria. And we have also discussed the functions of the important components, right? I do want to mention that some of the important functions, such as the role of mitochondria in programmed cell death, it is important in case of apoptosis. It is important in case of the liver cell detoxification process. It is also important in case of the proper maintenance of the calcium ion concentration. So there are a lot of research articles on this particular organelle, how it functions, and you can read those articles, and that will definitely improve your knowledge of this particular functions of the knowledge of the functions of this particular organelle. So I think that's it. We have designed our mitochondria, and we have also discussed the important functions of these individual components. I hope that this video was helpful for you to understand the mitochondria. I'll bring more topics in my future videos. I want you to stay tuned to the channel so that you can watch these videos and they're going to help you to understand the topic. All right, that's all for now. Thank you and take care.